is what a biological macromolecule is, and there are 4 types. So they are carbohydrates, lipids or fats, proteins, and nucleic acids. And so we're going to go ahead and we're going to talk a little bit about the basic concept of macromolecules and how they're used in biology, and then we're going to break those 4 types into separate videos. So I do want to talk about the fact that these carbohydrates, proteins, and nucleic acids are all what we call polymers, whereas lipids are not polymers. And that's important to remember. We've talked quite a bit about polymers and monomers in class. So a polymer is simply a long molecule that's made of similar or identical subunits, which we call monomers. So monomer, let's define that. So I've got my basically it's like a string of beads. So if I have a string of beads, the beads can be different colors, they can be different sizes, but they're all strung together in one long chain. And each of those is going to be held together by a covalent bond, and that's pretty important when we're talking about the biological molecules, that they're being held together by covalent bonds. Each of those beads, though, is going to be a monomer, so that's just going to be a building block or an individual subunit, and so that would just be the individual beads before I connect them, and that's really all we're talking about, a string of beads versus loose beads. That's polymer versus monomer, and so that's true for carbs, proteins, and nucleic acids. So let's talk about how we actually bond them together, because I've got these monomers and they're not covalently bonded to each other, and I need to somehow make them into the polymers, which are covalently bonded to each other. So I'm going to build or break down my macromolecules through one of 2 processes, and these should be familiar to you from chemistry. The first that we're going to cover is dehydration synthesis. Dehydration, synthesis. Synthesis means I'm building, and I'm building by removing water specifically. So I'm going to take a monomer, and then I'm going to take another monomer. And if you look at these monomers, when we're looking at these biological molecules, it's very common to have a hydroxide group, an OH, and then a hydrogen on an opposite side. So the way that I'm going to do this is I'm going to remove the hydroxide from one monomer, and then I'm going to remove the hydroxyl group from one monomer, and then the hydrogen from another. So I'm going to take those 2, and that's going to form HOH, which for those of you paying attention is water, and that's going to give me 2 monomers that need to form a bond. So they're going to bond to each other plus HOH. So that's my water, and there is the covalent bond that I've formed, and that's dehydration synthesis. The opposite of that would be adding water to break it down. That's hydrolysis, breaking down with water. Hydrolysis, break down with water. So I'm going to take my monomers, put them together into my polymer that's held together by my covalent bond, and then I'm going to add in some water. And so I've got my polymer, I've got my water, and what's going to happen is that polymer is going to have that HOH go in, and it's going to break that bond. I'm going to get a hydroxyl group on one and the hydrogen on the other, so that hydroxide goes and forms the hydroxyl. The hydrogen goes and attaches to normally a carbon, but not always. It could be a nitrogen, and I am going to get my individual monomers back out. So I've got my 2 monomers. Now this happens over and over and over again. So I could be talking about adding 2 monomers together. I could be talking about adding 2 monomers together and then individually adding like another 50 monomers. So I could have a chain of 51. Or I could be breaking down the whole chain. And these are the basic chemical reactions that we're going to be talking about throughout the entire school year. So it's really important that you narrowly an energy molecule. And they're made of carbon, hydrogen, and oxygen. And we use them for fast energy, and we also use them for some energy storage in plants and in animals. So they are structural components. Sometimes you do find some parts of cells that are made of carbohydrates rather than stored energy carbohydrates. Just a really quick example is a lot of the receptors on the outsides of cells that are used for cell recognition or recognition of signals are actually built of carbohydrates, and we'll talk more about that when we get to the cell membranes. But there's different examples that of carbs being used structurally in animal cells, and then obviously carbohydrates in plant cells are going to be used to make up that cell wall, right? And also in fungal cells. So let's talk a little bit about what makes up carbohydrates. Carbohydrates are monomers of monosaccharides. Mono, one, saccharide, sugar. So basically a carbohydrate is just a long chain of simple sugars. That's simple sugars. So let me draw you a really quick glucose. And for those of you that have been paying attention in class, this is alpha glucose. So that is an example of a simple sugar. That is a monosaccharide. C6H12O6 for those of you that don't remember. And we are going to go ahead and we're going to look at forming a polymer of my glucose molecules. So a polymer is also called a polysaccharide when we're talking about carbohydrates, and that's just going to be a large chain normally of glucose molecules or of other simple sugars. So here is my glucose, and I'm going to draw a couple. I'm just going to do one more. So we'll do another glucose. And those are both alpha glucose molecules. So I'm going to put them together, dehydration synthesis, remove the hydroxyl from one side and the hydrogen from the other. That's going to form a water. And then what I'm going to get is I'm going to get those two glucose monomers are going to join together. So let me go ahead and redraw those. And then I need to remove those groups. So let's erase that, and they just go ahead and they're going to both bond to that one oxygen that's remaining. And you'll notice that they formed that covalent bond in the down position, and that's what's going to happen consistently with alpha glucose. So with alpha glucose, you're going to just get a long linear chain, and we call that chain starch. So let's talk about some important disaccharides and polysaccharides. And when I say important disaccharides, we're really only going to talk about one important disaccharide, which is sucrose, because that's table sugar. There's a bunch of others that we'll talk about a little bit more when we get into fermentation, but really, I really care that you know sucrose. So sucrose is a disaccharide that is made up of glucose, and it is made up of fructose. Fructose, look at that, it's a pentagon. It looks like it's a 5-carbon sugar. Doesn't it look like it's a 5-carbon sugar? Except no, because there's a 6th carbon. Normally, where you've got that first hydroxyl group on the glucose molecule, if you look, there's a CH2OH on that side and on the left side. So on the 1 carbon and on the opposite side, you have that CH2OH. So I'm going to take both of those, that's my fructose molecule, and I'm going to do dehydration synthesis, and I'm going to form myself a sucrose molecule. Now, most of the disaccharides and polysaccharides that we're going to talk about are going to be used for storage and energy, although some are going to actually be structural. So let's talk the storage and energy polysaccharides first. The first one we've already talked about, it's starch. It's made by plants. There's 2 types of starch. We're only going to really concern ourselves with this one, which is amylose, and that's just a chain of alpha glucose molecules. The second one is glycogen, and that's actually produced by animals, so you make glycogen, and it's stored mostly in liver and muscle cells. So if you go out and you exercise, you're going to be using the glycogen that's stored in your liver and muscle cells. The other thing that glycogen is really important in is when you have a fight or flight reaction, you have a hormone, glucagon, that is going to go ahead and cause the glycogen to be released and broken down into sugar. That gives you quick sugar for that fight or flight reaction. So those are our storage molecules. Structural molecules are going to be cellulose. Cellulose is made by plants, and it is actually a polysaccharide of beta glucose. So instead of all of those bonds being down, what you're going to end up with is you're going to end up with them alternating. So you've got one goes up, one goes down, one goes up, and this creates a different shaped molecule, which is going to not be able to be broken down by the enzymes that you have in your body. And so it's hard to digest, whereas starch is easy to digest. And that's just because we have enzymes that will break down the amylose. We have amylase in your saliva. We don't have any cellulase. Cellulase is pretty much found in bacteria. And then the other structural polysaccharide is chitin. And chitin is actually found in arthropods, insects, and in fungi. In arthropods, it makes up the exoskeletons, and in fungi, it makes up the cell walls of the fungi. So that's that. I do want to talk about cellulose a little bit more. Cellulose, you're all very familiar with cellulose. Cellulose is the most abundant organic compound on Earth. And that's because that plants make up the majority of the biomass on Earth, and cellulose is actually it's the structural component of their cell walls. And cellulose is undigestible to most carnivores and omnivores. However, most herbivores actually can digest it, and it's because so cows can, but humans can't. And that's because that cows and most herbivores actually have a mutualistic relationship with bacteria and very long gastrointestinal tracts. So there's bacteria that live in their intestines, and they've got a very long intestinal tract, and that gives a lot of time as the cellulose passes through, or like in cows, they've got the 4 stomachs. It gives a lot of time for that digestion to occur. The longer the digestion takes, the more the bacteria can start to actually break down that cellulose. So you've got a mutualistic relationship between the herbivores and the bacteria. Now you have bacteria in your intestines that can break down the cellulose also. However, because you're an omnivore, your digestive tract is not as long. And so that's why there's at times that you'll eat something that's high in cellulose, like corn, and then you'll notice later that, oh, that didn't get completely digested. And that's just because our digestive tract is not long enough to provide the bacteria long enough time to completely break down that cellulose. So some organisms have those bacteria, and that aids in the digestion. And that's a mutualistic relationship. So the last thing I want to talk about on cellulose is that cellulose actually does have a role in human biology. Cellulose is also known to us as fiber. So in the back of your nutritional information, when you see fiber, and you know that fiber helps keep you regular, it helps those bowel movements. What fiber does, the cellulose, is it helps clean out undigested waste in your large intestine. And so that actually helps to prevent that waste product from building up in the nooks and crannies of your large intestine and your colon, and then leading to polyps, which can lead to cancer.
Let's talk about lipids. Now, for those of you who are familiar with the term, you might associate it with things like fat molecules, and that would not be incorrect. Fat molecules are a very common form of lipid. In fact, this is an example of a fat molecule, or a triglyceride right over here. Fat or triglyceride molecule right over here, and this one in particular is a polyunsaturated triglyceride, and we go into a lot of depth on this on the video on the molecular structure of fats / triglycerides, or on saturated and unsaturated fats. But you see the telltale signs. You see a glycerol backbone right over here, and 3 fatty acids, or what were 3 fatty acids, attached to what was a glycerol molecule. We go into a lot of depth on that, but fats are not the only type of lipids. And so what makes all of these other things that I'm about to show you also lipids? What commonality do they have with fats? Well, lipids are just the general term for a whole class of molecules that tend to not be so soluble in water, that tend to kind of clump up or ball up when placed in water. So not so soluble in water. And I'm being a little bit careful with my words. I didn't say outright hydrophobic. There are definitely lipids that are outright hydrophobic, but there are also some lipids that have some end of their molecule that's hydrophobic, but then other parts of the molecule actually might be hydrophilic. And there's actually words for that. So you have some lipids that are just straight out hydrophobic. They literally try to avoid the water. They're non-polar molecules, so they just clump together. But then there are some that have hydrophobic parts and hydrophilic parts. And these are called, and I always have trouble saying this word, amphipathic molecules, where some part is hydrophilic and some part is hydrophobic. And we're going to see that in a few seconds when we look at phospholipids, which are crucial for the structure of cell membranes. And we're going to see that a lot when you go into biology. So what are all these other molecules? And let's think about what parts of them might be hydrophobic and what parts might be hydrophilic. When you look at fats, you have this long hydrocarbon chain. There aren't any obvious charges over here. In fact, there aren't any, oxygen is more electronegative, so you might have a little bit of a partial negative or partial positive charge, a partial negative at the oxygen, or maybe a partial positive end at the carbon. But carbon is still more electronegative than, say, hydrogen. So you're not going to be able to form the type of polar bonds that, hydrogen bonds you would if you had hydroxyl groups here, if this was an alcohol. And these hydrocarbon chains over here, these are very hydrophobic. So that's what makes fat not be so soluble in water and clump up when you put it in water. This molecule right over here, which we would classify as an ester, and that's because we have an ester group. We have an ester functional group right over here where you have a carbon double bonded to an oxygen and then single bonded to another oxygen, and then that oxygen is bonded to a long hydrocarbon chain, and that carbon is bonded to a long hydrocarbon chain right over there. This is clearly going to be hydrophobic. And this particular molecule that we're looking at right over here, this is a major constituent of beeswax. Of beeswax. And if you've ever dealt with beeswax, or really any type of wax, and waxes in general are considered to be lipids, you can see that they're not soluble in water. In fact, they're often used to repel water, to keep water from penetrating into something. So wax, and in particular beeswax, and beeswax isn't made up of only this molecule. This is one of the main constituents. It has other molecules mixed in there, but this is also a lipid. Now, what's this thing over here? This thing, I have one 6-carbon ring, another 6-carbon. Whoops! I have another 6-carbon ring, another 6-carbon ring, and then I have a 5-carbon ring. This is the telltale sign. And let me circle these 4 rings right over here. These rings are the telltale signs that we are looking at a steroid. And in popular culture, steroids have, if you think of steroids as something that bodybuilders might inject illegally to pump up their muscles. But steroids are actually, when you think about it in chemical terms, they're actually referring to things that have these molecules that have this general structure where they have this 6-carbon ring, this 6-carbon ring, another 6-carbon ring in this orientation, and then another 5-carbon ring. And this steroid that we're looking at, this has an OH group attached to it, so it's actually going to be an alcohol. So a steroid that's an alcohol you would call a sterol. And this particular sterol is one that you've actually dealt with a lot, or at least you've heard about. This is cholesterol. So this one in particular is cholesterol. And cholesterol is often viewed as a negative thing. People want to lower their cholesterol, but it's essential for life. It's essential to the functioning of your cells, and it is a precursor molecule for your steroid hormones, which make you you. And this right over here is a steroid hormone. It's a very well-known one. This is testosterone. Testosterone. And you see the telltale, you have a 6-carbon ring right over here, another 6-carbon ring right over here. The double bond is in a different place than cholesterol. Here you have a double bond right over there. Then you have another 6-carbon ring, and then you have the 5-carbon ring. And instead of an OH group here, you have a double bond, you have a carbonyl group, you have a carbon double bonded to an oxygen, but it actually does still have an OH group up here. But this is a derivative of cholesterol. This is a testosterone. It is a steroid hormone. This is another steroid hormone. This is cortisol. Cortisol also can be derived from cholesterol. And you see the telltale signs. You see the 6-carbon ring. This has a double bond right over there. You see another 6-carbon ring. 6-carbon ring like this. It's actually hard to see the double bond, so I just won't even refer to them. I'll just refer to the general shape. So you have a 6-carbon ring there, 6-carbon ring there, 6-carbon ring there, and then 5-carbon, whoops, and then a 5-carbon ring just like that. So once again, they all have this steroid base structure, but then they also have other parts that make them different. For example, that is different than that is different than that. And just so you can visualize these things in 3 dimensions, in an actual molecule, they're not going to look exactly like this. And even talk about what something at an atomic scale looks like is kind of strange because light behaves in weird ways. But you can imagine the molecule would look like this if this is kind of a ball and stick model. Well, here you're thinking about what it actually might look like in space, where the white balls are hydrogen, the gray ones are carbon, and the red ones are oxygen. So this is also, this right over here is what a cortisol molecule would look like if you're thinking kind of a space-filling visualization. Now, I talked about amphipathic molecules, and phospholipids are probably the most well-known example of it. And phospholipids, they have a lot in common with triglycerides, in that you have this 3-carbon backbone right over here. 3-carbon backbone. Let me do that in a different color. So you have a 3-carbon backbone right over here. So this is a carbon, that's a carbon, that's a carbon there. Each of them are attached to oxygen, so you can imagine that this could have been derived from glycerol. And then 2 of the carbons, like in a triglyceride, are bound to a fatty acid like this. But then one of the carbons, the 3rd carbon, instead of being bound to a fatty acid like you have in a triglyceride, is bounded to a phosphate group. So this right over here is a phosphate group, and this R could just be another chain, another organic chain, so to speak. But when we talked about amphipathic molecules, I always have to say it slowly, so it's a bit of a tongue twister for me. We're talking about having a hydrophobic end and a hydrophilic end. Well, what's the hydrophobic end here? Well, this chains from the fatty acids, especially the hydrocarbon chains right over here, these are going to be hydrophobic. Hydrophobic. While the phosphate end right over here, it has charge. Charged molecules dissolve in water very, very well. And so this one over here is going to be hydrophilic. It's going to be, I guess you could say it's going to be attracted to water. And that's why phospholipids, and this is just one type of phospholipids, these chains could be different. We have a unsaturated chain here, and then a fatty acid chain, and then we have a saturated fatty acid chain on the left. And this is just, I actually just made up this molecule, and they could be different depending on the phospholipid you're talking about. But this general property of having a hydrophilic head and hydrophobic tails make them very well suited for cellular membranes. Because you can imagine, so that end is hydrophilic, and then you have these hydrophobic tails. And actually, let me see if I can copy and paste this really fast. I actually can just draw it really fast. So let me just draw a bunch of them. Whoops. Nope. I'm having trouble. All right, let me just switch back to my drawing tool. So let me just draw a bunch of the hydrophilic heads, and then a bunch of the hydrophobic, actually I'll draw a few more hydrophilic heads out here, and then I'll draw all the hydrophobic tails. Hydrophobic tails, let me draw them really fast. I'm almost done right over here. Hydrophobic tails right over here. And this configuration that I've just drawn, where you have a bilayer of phospholipid, a phospholipid bilayer, this is how cellular membranes are constructed. Because this, you have water, or things that are very water-based, inside the cell. This could be inside the cell, and this could be outside. This is outside the cell. And so the phosphate ends is attracted to the water, and then the hydrophobic, the hydrocarbon tails, those are going to they're going to orient themselves in this way to get away from the water, really to just let the phosphate ends interact with the water. And so this forms a nice boundary for the cell, and we're going to study that thoroughly as we go more into biology. So hopefully this gives you more appreciation of what a lipid is and the different types of lipids.
Proteins are the most structurally and functionally diverse group of macromolecules, and they're actually the group of macromolecules that we're going to spend the most time talking about this year because they're pretty much involved in everything. So let's talk about the different functions that proteins can have. So first off, all the enzymes that we're going to talk about this year, enzymes are proteins. So an enzyme, we're going to have a whole separate set of notes on enzymes, but enzymes are proteins, okay? So an enzyme is a protein. It's got a specific shape. It does a specific job, either building something or breaking something down. The examples that we're going to talk about the most in class are catalase, DNA polymerase, lactase, because I'm lactose intolerant, and ATP synthase. Now, what do you notice about all of those enzymes? Well, they all end in ase. And that's how we know that something's an enzyme. So proteins that end in ase are enzymes, and normally their name tells you something about what they do, like lactase breaks down lactose sugar. So that's enzymes. The next thing that we need to talk about with proteins is that they are structural. They provide structure to the body. So collagen, keratin, just to name a few. So like your hair, like the filler proteins that actually provide shape to your cells, those are all proteins. Microtubules, microfilaments, and your cytoskeleton, those are proteins. So those are structural. Proteins are also involved in the transportation of things on an organismal level and on a cellular level. So on an organismal level, hemoglobin is how we transport oxygen around the body. On a cellular level, protein channels allow certain molecules to move through the cell membrane. So you all learned about protein channels in the cell membrane when you took Bio 1. So that's involved in transport. We also need to talk about cell-to-cell communication. Now, we haven't talked at all about this in class, but cell-to-cell communication is how one cell or a molecule outside a cell is going to go ahead and trigger a reaction in another cell. So in cell communication, you're going to have a signal molecule, and that signal molecule could in fact be a protein. So hormones like insulin is a hormone that is actually a protein hormone, and it triggers a response by the body, right? That's a signal molecule. But you could also have receptors that are proteins, and we'll talk about specific receptors on the cell membrane, inside the cell, that are floating in the cytoplasm, that are receptors that trigger different pathways in metabolism. And then we also need to talk about antibodies. We're going to do that. That's a whole separate unit. So antibodies, the way that your body actually defends itself against pathogens, like it recognizes specific antigens and produces antibodies. Well, those antibodies are basically protein tags. And so you have to be able to make those proteins. And then movement. Obviously, your muscle cells, they're going to be made of actin and myosin. You're going to have actin and myosin fibers, and those actin and myosin fibers are proteins. So there's a ton of different structurally and functionally different proteins, and we're going to talk about a lot of them. So let's talk about how a protein is actually built. So we're going to build proteins by taking monomers and forming what we call a polypeptide. So a polymer, the protein polymer, is called a polypeptide, and we'll talk about why it's called a polypeptide in a second. The monomers of a protein are amino acids, right? And there are 20 amino acids, and we can make a lot of the amino acids, but we can't make all of them. So most of these amino acids you're getting through your diet. Some of them you're taking in certain amino acids and then you're synthesizing them. Amino acids have an amine group and they have a carboxyl group. And let's go ahead and review that, even though we've done it together already in class. So this is the structure of a generic amino acid. And yes, I am missing a bond there. I'm aware of that. So my amine group is this NH2 on the left-hand side, and my carboxyl group is this C double bond OOH on the right-hand side. So that is my amino acid. That's why we call it that. Now this is a polar molecule, Okay, the amine group carries a positive charge, and the carboxyl group is negatively charged. So, this is going to go ahead and create some interactions between them. So let's look at the missing bond, because I have a missing bond, right? So that is going to be my functional group, and I represent the functional group with just the letter R. I will try and always circle the functional group when we're talking about it generically, so you know that it's not, there's no atom on the periodic table that's an R. So R, there's my functional group, right? So that's a generic amino acid. The only thing that's different between the 20 different amino acids is what is hanging off in that R position. So let's look at an example of actually taking two amino acids and putting them together. So I'm gonna form a polypeptide. I'm gonna do this by taking two monomers, putting them together, removing a water, dehydration, synthesis, right? So I'm gonna take my amino acid, and this one, if you notice, my functional group is just an H, so that's glycine. And I'm going to add to that this amino acid, and here I have a CH3, and that is an alanine. Okay, so I'm going to take my glycine and my alanine, I'm going to remove a water, and then I'm going to take that extra carbon covalent bond, because carbon needs to form a covalent bond there, and it's going to actually bind to that nitrogen where I removed the hydrogen. And that is what I call a peptide linkage. That's the type of bond it is, right? So when I say that a protein is a polymer, is called a polypeptide, it's called a polypeptide because basically I'm taking an amino acid and I'm hooking it to another one through a peptide linkage, so it's a polypeptide. So that's how you're going to build your protein chains, right? We're all familiar with those. That is your hereditary material. It's your hereditary information. It's on your chromosomes. It's in your genes, whatever you want to say about it. It's your nucleic acids, right? And they're made of monomers of nucleotides. All right, so DNA is made up of 4 nucleotides: adenine, thymine, guanine, and cytosine, and then RNA is made up of A, G, C, and U, and we're going to review that again in a second. So let's look at what a nucleotide is first. So a nucleotide is made up of a sugar to start. So we have a sugar, and in DNA, our sugar is deoxyribose, which is a 5-carbon sugar. In RNA, our sugar is ribose. So let's look at this 5-carbon sugar, and then we're going to go ahead and we're going to attach it to a phosphate group. Now, because that phosphate is negatively charged, this is going to provide an overall negative charge to my DNA, which means DNA is most definitely charged, which means it is most definitely going to be hydrophilic. So it's going to like water. So I'm going to take my phosphate group, attach it to a sugar, and then it's going to attach to the working end, which is the nitrogen base. And this is a super generic nitrogen base. That's a made-up nitrogen base. Don't think there's one that looks like that. So that's my nitrogen base. Okay? And so that, all 3 of those together make up a nucleotide. When we did these in class, I just had you make the base because we didn't have large enough kits to make the sugar and the phosphate group too. So let's go ahead and let's look at the basic structure of DNA. So DNA, you have a yellow sugar, or you're going to have a yellow phosphate group attached to a white sugar, right? So yellow, white, yellow, white, yellow, white. So basically what I have is I have this backbone going down the sides that's phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And then hanging off the sides, you end up getting the nitrogen bases. And the nitrogen bases, because DNA is 2-stranded, right? The nitrogen bases stick off towards each other and they form like the rungs of a ladder, and they bind together through hydrogen bonding. All of the bonds in what I've got drawn here are covalent bonds, but when I actually have like A and T next to each other, they're going to form hydrogen bonds. Okay, they're not going to actually form a covalent bond across the strands. They form they actually form many hydrogen bonds. So that's my DNA structure. That's a single-stranded structure, but we made it DNA. It's a single-stranded DNA. So let's talk about the 5 types of nucleotides. So my 5 nitrogen bases, there's only 5 nitrogen bases. There's adenine, which always pairs to thymine in DNA, and then guanine, which always pairs to cytosine in DNA. And then in RNA, I have uracil, okay? So adenine and guanine are what we call purines, which basically just means that they're double ring structures. So see the double ring structures below? Like, don't memorize them. That's a waste of your energy. But they're double ring structures. Adenine and guanine are double rings. Thymine and cytosine and uracil are what we call pyrimidines. And I always remember this because uracil is the shortest of the nucleotides, and it's a single ring. So single ring, but it's the long word. Pyrimidines sounds like it should be that double ring thing, and it's not. So I just remember that I'm wrong all the time, and that the shortest word, uracil, is a single ring and goes with like the long pyramid type thing. That's how I remember it. I don't know how you're going to remember it, but you need to know that. So uracil is going to bind to adenine. So if I have an adenine in my DNA, when I make an RNA copy of that, it would go A to U. And we're going to do activities on that in class. So those are my 5 nitrogen bases. And so in DNA, I'm going to have a double strand, and I'm going to have A, T, G, and C. So I'm going to put like a little yellow check mark next to all 4 of those. And then in RNA is single-stranded, and I'm going to have A, U, G, and C. So I'm going to put a pink check mark next to A, U, G, and C. And so that's your basics on the monomers of nucleic acids.